Okay, so question 2. I've read conflicting statements about the absorption of monomers, saying that they diffuse in with sodium ions in the co-transport, or others saying that it's active transport. Please, can you clear this up for me? What is co-transport? So let's just have a look at the diagram first of all. So this area here is the lumen of the gut. These here are those microvilli cells that make up the kind of edge of the villi in the ileum. And down here, we would have the capillaries. And in between those two areas, you would have the tissue fluid. So we've got 3 protein kind of channels that are in play. We've got this one up here. Now it's this one up here that is actually the co-transport protein, because it's co-transporting either the glucose and the amino acid with the sodium ion. And then down here, we've got one that's acting as a pump. So this is doing active transport because it's using ATP, and that's doing active transport of the sodium ions. And then over here, we have got a protein that's allowing the facilitated diffusion of that glucose or the amino acid. Now, the first thing to point out is that you can't actually actively transport glucose or amino acids. This is the system, this co-transport system is the system that has evolved to allow these molecules, the glucose and amino acids, to be taken into the cell, regardless really of their concentration gradient. So this co-transport will bring every last little bit of glucose and amino acids into the cell here by facilitated diffusion. So how does it work? Well, actually, we're going to start off at the bottom here, down with this protein pump here that's using ATP, because the first thing that we need to think about is that so the sodium ions are actively transported out of the microvilli cell using ATP. So the sodium ions that are coming in, they are actively pumped out into the tissue fluid. And this ensures that there is always a lower concentration inside the microvilli cell compared to here in the lumen of the intestines. Now that is important because it means that sodium ions are always going to be able to enter into the microvilli cell down their concentration gradient. So because that concentration gradient has now been set up, because the sodium ions are getting actively transported out of this kind of second, kind of the other end of the cell, it means now that these sodium ions are going to come in through here, and they're going to enter, like I say, down their concentration gradient. And when they do, they will bring in with them, they'll co-transport the glucose or the amino acids with them. So they will enter into the cell with those sodium ions. Once the glucose or amino acid are then into the cell, then because they've been brought in, the concentration of those is now going to be higher here. The blood is constantly moving, so there's going to be a low concentration of those molecules in the blood. And therefore, now what's going to happen is the glucose and amino acid molecules are going to be transported out of this microvilli cell into the capillary by facilitated diffusion. So sometimes this is called secondary active transport because this process must be doing the active transport of those sodium ions to allow the co-transport here molecule to be used. But just let me make it abundantly clear that this is not active. So the co-transport protein is using facilitated diffusion to bring the sodium ions and that glucose molecule in.